Previous case studies established that adult telomerase-positive stem cells, or aTPSCs, reside in a quiescent state, capable of activating and dividing symmetrically to repair damaged tissue. The objective of this review is to examine the exact clinical efficacy of these specific cells when deployed against severe, refractory autoimmune conditions that entirely fail to respond to standard medical care. The clinical subject for this intervention is a 61-year-old male. He presented with a severe, genetically inherited constellation of autoimmune disorders, most notably a case of refractory celiac disease that had persisted despite strict dietary compliance. This line graph maps the primary diagnostic biomarker for celiac severity, the deaminated gliadin peptide antibody titer. A measurement below the 1.0 threshold indicates no allergic reaction. Anything above 1.0 confirms the presence of the allergy, with higher numbers indicating more severe mucosal inflammation. To understand the severity of this patient's condition, we have to look at the testing laboratory's historical baseline. Prior to this case, the highest reaction the lab had ever recorded was a score of 37. The patient's baseline antibody titer registered at 73. His immune intolerance to gluten was nearly double the worst case the laboratory had ever documented. By utilizing a highly targeted multi-lineage stem cell protocol, clinical researchers safely reduced this extreme titer to 0, granting the patient 8 consecutive years of clinical remission. Overriding an autoimmune response of this magnitude required a combined protocol. The clinical team utilized both autologous stem cells, harvested from the patient, and allogeneic stem cells, harvested from healthy donors, to maximize the regenerative effect. This matrix details the exact stem cell lineages evaluated for the treatment. Adult stem cells fall into distinct categories, including totipotent, pluripotent, and mesodermal cells. For the autologous portion of the treatment, the team could safely extract, activate, and reinfuse all 3 of the patient's own cell lineages, totipotent, pluripotent, and mesodermal. The allogeneic donor pool, however, required strict filtering. The clinicians only utilized donor totipotent and pluripotent cells. Donor mesodermal stem cells were absolutely excluded from the treatment. The determining factor for this exclusion is listed in the final column. Mesodermal cells test positive for MHC class I expression. MHC class I markers are surface receptor proteins. They function as a unique biological ID tag, allowing the body to distinguish self from foreign material. Understanding and isolating these specific surface markers is the strict biological prerequisite for safely introducing donor stem cells into a highly reactive autoimmune patient. When an intact immune system encounters foreign MHC class I markers, it initiates a relentless search and destroy mission to eradicate the non-self entities. If the clinical team had infused allogenic mesodermal cells, they would have faced 2 potential failures. The first is immediate rejection. The host's hyperactive immune system identifies the donor meso-CSCs as foreign and destroys them, rendering the therapy useless. The second possibility is far more catastrophic, stemming from the fact that mesodermal stem cells possess the capability to differentiate into immune cells. If the donor meso-CSCs survived and differentiated, they could generate a completely new, foreign immune system inside the patient's body. That new immune system would read the patient's own tissue as the enemy and begin attacking the host organs. In clinical immunology, this fatal conflict is defined as graft versus host disease. Totipotent and pluripotent cells solve this problem. Because they natively lack MHC class I markers in their undifferentiated state, they are effectively invisible to the host's immune defense. By strictly filtering out the mesodermal lineage, the clinical team harnessed the regenerative power of the allogeneic donors while entirely bypassing graft versus host disease. The intervention took place over a rigorous 8-year period, utilizing regular infusions of the patient's autologous stem cells. This was supplemented by a highly controlled allogeneic protocol. Over the entire 8-year span, the patient received exactly 9 allogeneic stem cell donations. These 9 donations were sourced from 4 distinct donors, ranging in age from 42 to 80. Every donor underwent extensive genetic sequencing to verify they were completely free of celiac disease and any other autoimmune genetic mutations. Returning to the clinical data, we see the results of this combined protocol mapped across the 8-year window, marked by the 9 donor infusions. The patient's antibody titer crashed from the peak of 73 down to a complete flatline, resting securely below the 1.0 threshold for the entire duration of the therapy. In serological terms, the inflammation was gone. In functional terms, the patient's severe allergy was eliminated. The patient resumed a completely normal diet. He consumed gluten, barley, and rye, and remained totally asymptomatic as long as the treatments continued. This leads to the long-term reality of the intervention. A sustained drop in antibody titers proves the therapy manages the pathology, but the question remains whether this constitutes a permanent genomic cure. After 8 years of successful symptom management, the clinical team halted the allogeneic stem cell treatments. Looking at the follow-up data, approximately 1 year after the donor infusion stopped, the patient's immunological tolerance began to break down. The deaminated gliadin peptide titer spiked aggressively, crossing the 50 mark and trending back toward his original baseline. The physical consequences mirrored the serology. The severe clinical symptoms of celiac disease returned, and the patient was forced to abandon his normal diet and reinstate strict gluten-free restrictions. Ex vivo activated telomerase-positive stem cells possess profound immunomodulatory power, capable of overriding severe autoimmune pathology for nearly a decade. However, for advanced genetically rooted refractory cases, continuous or periodic stem cell maintenance is required to sustain mucosal tolerance.